Dear students, Assalamu alaikum. Today I am going to deliver a lesson for class 12 subject zoology. In our previous topic we discussed about mutation and causes of mutation. Now today we will discuss about the types of mutation. Mutation are broadly classified into two types. Number one is the chromosomal mutation and number second is the genetic or point mutation. So for the chromosomal mutations are concerned, they are further categorized into two types. First are those changes which are caused due to the change in the structure of chromosome, are also known as morphological mutations. And second type of chromosomal mutation is what we call as a numeric mutation, in which there occurs change in the number of chromosomes. Now, so far the morphological mutations are concerned, they are of different types. For example, deletion, duplication, inversion, and translocation. Now, so far deletion is concerned, here a loss of the segment of chromosome is caused initially by a double break in the chromosome and followed by a reunion of the segment. For example, in this case, the segment FG is lost in this case, and in this chromosome, the resulting chromosome, there is missing FG part. This is the example of deletion. Now, second example of this morphological mutation is the duplication. In duplication, one extra chromosomal segment is attached to the normal homologous chromosomes. In this case, we see that this FGH segment is first broken, then it gets reunited and gets duplicated, in this case EFG, and H. Then the third type is inversion. Inversion is a type of chromosomal aberration in which a part of the chromosomal segment gets inverted by 180 degree. For example, here in this case, the chromosome having the genes A, B, C, D, E, F, G, H, I develops inversion in the part FGH to form A, B, C, D, E. Then the inverted segment, instead of this F, we get HGF. This is the example of inversion. Now, another example of this is this chromosomal morphological mutation is the translocation. Here, separation of a chromosome segment takes place. Then broken segment is attached to the non-homologous chromosome, as is seen in this sketch. Now, the second type of chromosomal mutations are called as numeric mutation, in which the change in the number of chromosomes occurs. Now, it is further classified into two types, that is euploidy and aneuploidy. In euploidy, the change in chromosome occurs by a set, whereas in aneuploidy, change in chromosome number occurs by 1 or 2, that is plus minus 1 or plus minus 2. So for euploidy is concerned, it is of different types, such as haploidy, in which the chromosome number is changed by one set, that is represented by N, and we see this condition in case of gametes formed at the time of meiosis. Then is the diploidy. This condition is represented by 2N. It indicates two sets of chromosomes, and this condition occurs in normal somatic cells. Then there is triploidy. In triploidy, there are three sets of chromosomes represented by 3N, and this condition is seen in endosperm tissue in endospermic plants. Then we have the fourth type of euploidy, known as polyploidy. Now, polyploidy is a phenomena of having more than two sets of chromosomes or genomes. Polyploidy occurs in nature due to the failure of chromosome to separate at the time of anaphase, either due to the non-disjunction or non-formation of spindle. Now, it can be artificially induced by application of colchicine or certain other drugs. An organism or its karyotype having more than two genomes is called a polyploid. Depending upon the number of genomes present in a polyploid, it is known as triploid, tetraploid, pentaploid, or hexaploid, etc. Now, the second type of numeric mutation is what we call as aneuploidy, where the change in chromosomes occurs by 1 or 2, that is plus minus 1 or plus minus 2. It occurs in different forms. The different forms of aneuploidy are monosomy, nullisomy, trisomy, and tetrasomy. So far monosomy is concerned, it is represented by 2n-1. It is an aneuploid in which one chromosome is devoid of its homologue. Monosomic is generally weaker than the normal form. Turner syndrome is an example of monosomy. Turner syndrome is the monosomy in case of sex chromosomes, where the condition can be represented as 44 plus X. We see the Y chromosome or another X chromosome is missing here. This is the Turner's syndrome and represents the monosomy. Now the second type is nullisomy. Nullisomy condition is represented by 2n-2. These nullisomics usually do not survive except in polyploids. Here the aneuploid is deficient in complete pair of homologous chromosome. Then the trisomy. Trisomy, as is seen in Downer syndrome, the condition represented by 2n+1. For example, in Turner syndrome, there is trisomy of 21st chromosome. Then the tetrasomy. The tetrasomy condition represented by 2n+2. This type of this aneuploid is seen in apple and pear. They show more variability because of tetrasomy, and because of this tetrasomy, they give rise to new species through secondary polyploidy. Now, this was all about the chromosomal mutation. Now we will discuss about a little bit about the gene or point mutation. Now, change in the expression of a gene caused by change in number, sequence, and types of nucleotides is what we call as gene or point mutation. Now, if the gene mutation occurs due to change in a single base pair of DNA, it is known as point mutation. This gene mutation occurs by three types. The first is inversion, where we see here the base pair gets inverted, and they form new codons which give rise to different types of amino acids. For example, GCC and TTG, when inverted, they form GTT and CCG instead of TTG and GCC. So different codons code for different amino acids. Thus, they manipulate the genetic information. The second type of genetic or gene mutation is substitution. Here, the replacement of one or more base pairs occurs. For example, transition. It occurs by transition. That is, pair is replaced by another pair. That is, for example, the purine A is replaced by purine guanine, and transversion. In case of transversion, a purine is replaced by a pyrimidine. Then we have the last type of this gene mutation, what we call as frame shift mutation. Now, in case of frame shift mutation, insertion or deletion of a base pair in DNA occurs. This shifts the reading frame of codon from the site of insertion or deletion. Now, muscular dystrophy in case of humans is a very famous example of frame shift mutation. Now, this was all about the types of mutations. In our next lesson, we will discuss about the importance of mutation. Until then, goodbye.